Alright, now for the last section of Structure and Function of Biomolecules, Part 3, Proteins. Proteins have many different functions. There are thousands of different proteins in cells to do lots of different things in organisms. Some proteins are used for communication. Insulin is an example. Proteins will be enzymes. They can be antibodies and used for defense in animals. They can be receptors and receive signals. They can be structural to support a structure. They can be motor and contractile. And they can be energy or storage, like in a seed. The structure of amino acids begins with the amino group. It is going to be in its NH3 plus ionized form in a cell. The carboxyl group will also be in its ionized form, and they have a hydrogen and an R group. This R group can be variable. That means it has different properties due to the different functional groups. We'll look at those in just a minute. Polypeptides are going to be joined by a peptide bond between two amino acids. This is formed by dehydration synthesis, and it's formed between the carboxyl of one and the amino of another. This gives the protein direction. The protein backbone will have the pattern NCC, NCC, with the peptide bond between the two amino acids. The N-terminal end would usually be the beginning of a polypeptide chain. The carboxyl terminal end would be the end of a polypeptide chain. There are 20 different amino acids. The R groups are highlighted in blue. These 20 amino acids are responsible for all the proteins and all their different structures. These 20 amino acids are found in all organisms, and it's the R groups that are responsible for the protein structure, and it's due to their properties. Some are nonpolar, some are polar, some are charged, and some have an aromatic ring or a ring structure. These different shapes and structures will give these amino acids various properties and help shape the protein. So this leads to infinite variety. That's why we can have thousands and thousands of different proteins to do lots of different cellular processes. So how do we get a shape of a protein? Well, it all starts with its primary structure. And the primary structure is strictly the order of the amino acids as instructed by the gene. There is the central dogma that the information begins with the DNA, which is then converted to RNA, which is then converted to protein. It is this information process that gives us the primary structure. Once we have the primary structure, then we move to the secondary structure. This is how the amino and carboxylic ends of the amino acids interact by hydrogen bonding. Notice the NCC pattern of the amino acid's backbone. There are two kinds of secondary structure. You can form an alpha helix. This is with every 4 amino acids hydrogen bonding together, and this will form this spiral-based structure. Then the other structure you can have is the beta pleated sheet. This is when you have hydrogen bonding between segments of the backbone that are parallel to each other. This gives a flattened region of the polypeptide chain. Now, proteins can have one type or both type of secondary structures in the overall structure of the protein. Keep in mind, hydrogen bonds between the positive amino and negative carboxylic ends of the monomers. That is what causes secondary structure. For tertiary structure, it is the R group interactions. There are lots of different interactions that are going to give us a three-dimensional shape to the protein. And this shape will always be the same for each individual protein because it's based upon the order of the amino acids. You can have ionic bonding between the charged R groups. You can have hydrophobic interactions between nonpolar groups. You can have disulfide linkages between sulfhydryls, and you can have hydrogen bonds between the polar R groups. It is critical to understand that this three-dimensional shape would be the same for each individual protein. Shape is going to dictate the protein's function. Some proteins are made up of multiple polypeptide subunits before they are going to be functional. These are going to be weak interactions and other interactions, just like in tertiary structures. An example of a protein is catalase. Catalase is going to have 4 identical subunits. This is catalase. This is the ribbon structure for catalase. Notice the helices of the alpha subunits and the beta flatted regions of the beta pleated sheets. This would be an example of a protein with a quaternary structure. Protein's shape is critical to its function. If there is a mistake in the DNA, which causes an amino acid substitution, like with sickle cell anemia, the normal hemoglobin, glutamic acid, is going to be replaced with a valine. This is only one amino acid in the entire sequence that is changed. Because of this change, the hemoglobin is no longer shaped properly. It is rigid, it is in strands instead of a globular formation. So basically what happens is we replace a hydrophilic amino acid with a hydrophobic amino acid, which gives the hemoglobin a different shape. The sickle cell hemoglobin no longer transports oxygen efficiently. So just a change in one amino acid in one R group causes a change in the shape of the protein and its function. So proper protein shape is critical to how biological processes work in the cell. This concludes our section on macromolecules.